So hello, ladies and gentlemen. I'm Dr. Shubar, coming from University of Tripoli, Faculty of Pharmacy, and I'm working as an assistant professor. I did my master's and my PhD in Berlin in the Charité Medical School. And at this moment, I'm presenting my master work. So my talk will be about the role of apolipoprotein E in the uptake of autovacone into the brain, murine, and acute and reactivated toxoplasmosis. Just briefly, what I'm doing here is we have a model of toxoplasmosis. As we know, Toxoplasma gondii is a parasite that a person could be infected orally. It goes to the oral cavity. It distributes all over the body organs, and then finally the last stage will be the brain. So the cyst will persist in the brain lifelong, as soon as or as far as your immune system is working well. So that means if there is any defect in the immune system, this parasite the parasite cyst will rupture, and then this will lead to toxoplasmic encephalitis. So I have two models here. I have the acute model and I have the reactivated model. The drug of choice for the treatment of toxoplasma or toxoplasmosis is the autovacone. The problem of this drug is it has a low bioavailability, and that's why what is present in the market is present in the micronized form. So here in the Berlin Medical School, we synthesize the same drug but in nanoparticles in order to enhance the uptake and to enhance the solubility of the drug. Furthermore, I coated the nanoparticles with surfactants. I used poloxamides, and I used tween 80 as a surfactant for the wettability of the drug also. Furthermore, I also coated the nanoparticles with apolipoprotein in order to see whether the drug is taken into the brain via the ApoE pathway, because as we know, apolipoprotein transfers normally the LDL to the brain by binding to the LDL receptor. So I'm just investigating whether the autovacone is taken into the brain by the same pathway. So what we did as I told you, I synthesized the nanoparticles. I did the coating with tween 80 and other surfactants, plus also I did the coating with apolipoprotein peptides, and also the size ranges from 200 to 400 nanometers. And this was the method of production. So I have, as I said, I have the acute model of toxoplasmosis, or acute murine model of toxoplasmosis. I used the ApoE knockout and the control black 6 mice. We treated the mice with 10 cyst ME49, the chronic form of the parasite. And then on day 9, 12, and 15 post-infection, we treated the mice with the nanoparticles or nanosuspensions, 10 milligram per kilogram, administered as a single IV dose. And then one day after the last dose of acute IV therapy, that means 16 days post-infection, sera, brain were obtained for histological and pharmacological analysis. This was the reactivated model. The reactivated model, just briefly, these mice are knockout mice. They lack the interferon regulatory factor 8, which is responsible for the resistance against the parasite. That means this is like a model that resembles people that have a defect in the immune system, for example, like HIV, people like receiving organ transplantation, and so on. So these interferon regulatory factor 8 knockout are infected with the 10 cyst ME49 toxoplasma, and then 2 days post-infection, I have to treat the mice with sulfadiazine in drinking water in order to enhance the latent infection and the cyst formation. That means if I don't treat them with sulfadiazine, the mice will die within 3 days. So then I stopped sulfadiazine after 28 days. 2 days after stopping sulfadiazine, I start my acute IV therapy on day 2, 5, 8 after discontinuation of sulfadiazine, and then at day 9 after discontinuation, which is 1 day after the last IV therapy, the same thing, I collect the organs for HBLC, PCR, and histological analysis. This was the data of blood concentration in serum and brain measured by HBLC. This was the reactivated model. in interferon regulatory factor 8 knockout. As we can see here, I have the four different formulation, otavocone twin 80 with or without ApoE. The same thing with poloxamir 184 with or without. The same thing with poloxamir 338. In conclusion, we can see that there is no significant difference between the drug concentrations between each formulation. That means the drug is available for the whole formulations. The same thing in the brain. For the acute model, I used the wild type, the black 6, and the ApoA knockout. And this is the concentration also in serum and brain. And as we can see, the black bars here is the drug without apolipoprotein, and the white bar is without apolipoprotein. Sorry. So the black bars are with apolipoprotein, and the white bars without apolipoprotein. So which is more important to me is the ApoA knockout. That means here in the white bar, when there is absence of apolipoprotein in the mice and absence of the apolipoprotein in the formulation, I still get drug concentration of the, I still get drug in the serum. That means the drug is there in the serum and in the brain in the absence of both apolipoprotein. And this is also regarding the parasite numbers. I used to investigate the parasite numbers by PAP, which is the peroxidase antiperoxidase staining technique. And here the parasite number, as we see here, uncontrolled also, of course, will show a high number of parasite. Comparing it with the treated groups, the number of parasite was significantly reduced in both formulations. And also here in the ApoE knockout, which is very important to me. That means in the absence of ApoE in the formulation and absence of ApoE in the mice, I still get an effect of the drug. That means ApoE doesn't play any role in the uptake of the drug into the brain and orally. Furthermore, we coated our ApoE, our particles with the ApoE covered with the green fluorescent or green fluorescence coated ApoE. And what we did here, we incubated BN3 cell line, which is a brain cell line. We incubated for 30 minutes with our particles, and then we used confocal laser scanning microscopy to detect the appearance of the nanoparticles on the cells. And as we can see here, this is only the accumulation of the particles on the cell rather than internalizing the particle inside the cells. And this also we stain the protein also with different color, the green and the red, and the same thing as we can see here, the particles are not internalized inside the cells rather than just accumulating on the cell surface. And this really will correlates our results or matches with our HPLC and histological results. As a conclusion, we can say that the addition of apolipoprotein did not increase the uptake of otavocone nanosuspensions into the brain in vivo. Neither the addition of ApoE peptides to the anus nor naturally occurring ApoE contributes to the uptake of nanoparticles into the brain and the therapeutic effect of otavocone. ApoE or apolipoprotein appears to contribute to the accumulation of the nanoparticles on the surface of the brain endothelial cells, but not in their uptake into the cells. And finally, of course, the work was done by different departments or different institutes in Berlin. So Institute of Microbiology and Hygiene, Charité, Campus Benjamin Franklin, Leipzig Institute for Molecular Pharmacology, Department of Pharmaceutical Technology and Biotechnology, Free University Berlin, and Institute of Clinical Chemistry and Pathochemistry in Berlin, Campus Benjamin Franklin. Thank you very much.
Thank you very much.